Hi everyone, it's Kim here today, and today we are talking about methods to quantify wildlife. So let's first talk about our learning objectives. When we're talking about quantifying and measuring wildlife, the first thing we want to understand is its importance when it comes to planning. We need to understand what it is we're quantifying or what it is we're measuring before we even set out to do it. We'll also be talking today about measurements as they're related to the populations and overall population management, but we will also discuss measurements related to the habitat and habitat management. So let's just start at the beginning. Why is it important to quantify wildlife? There are three categories under which knowing the number of animals is helpful, and the first is to assess environmental conditions. If we know a bit about the animals, and if they're there or are not in the area and can relate that to an environmental characteristic, it allows us to create better management. So for example, we can look at a study of black bears in New England. Researchers wanted to know how the probability of occurrence of black bears was influenced by the amount of development nearby. So to answer that question, they looked at a number of black bear sites in areas of high, medium, and low levels of land development. Researchers ended up finding that the amount of development within 5 kilometers of the site, if that increased, there was a lower probability of occurrence of black bears. And so in this case, it relates directly to landscape conditions because it came down to where black bears could find suitable habitat. So again, this comes back to the environmental conditions of what is suitable habitat for those black bears. We can also talk about how many can be harvested. That's the second point under which knowing how many animals are available or are there becomes helpful, because if we know how many, we have the ability to model and understand surplus, or basically the quote-unquote extra and the amount that may be available for harvesting. And finally, by understanding those relationships in terms of environmental conditions and also how many may be harvested or may be extra in the area, we can then anticipate and quantify how much environmental change may influence the species. So in other words, we can look at a population's resiliency. And you'll hear this word come up pretty frequently when you're looking at the ecology of a particular species. So again, let's go back to that black bear example. If we're in a situation where development continues to increase and there becomes more and more land that is developed within 5 kilometers of where we typically see black bears, we can see and project that our probabilities for black bears to occur will become smaller and smaller. We'll see less and less black bears in the area. But let's also talk about things like, what is it that we want to quantify in wildlife beyond simply are they there or are they not? And also, what about demographic features like births and deaths or immigration and emigration? We will also spend some time discussing how we quantify habitat via occupancy, selection, and behavior. So occupancy is basically, are they there or are they not? Selection is, are they choosing one area over another? And behaviors are how they're choosing to move around those areas that you do find them. And there can be so many ways in which we can go about collecting this data. So for instance, each fall at Hawk Mountain Sanctuary in eastern Pennsylvania, interns and wildlife professionals will actually spread themselves across ridges in the area to complete a raptor count during the annual raptor migration. And so these individuals are taught how to ID birds of prey by sight and by sound so that the observers can count how many individuals and species pass through the area. So you can see this would be a pretty successful approach in terms of being able to both count those birds and understand which birds it is that they're hearing. And you don't necessarily just have to see the organism, as we were saying in that bird example, in the raptor example. You can also go about this with acoustic measurements, which is actually how we end up categorizing or counting wildlife like bats and birds and frogs. And so there are many, many ways we can go about collecting some of these basic numbers, whether it be the population numbers or via habitat use. And so ultimately what we need to know is a little bit more about the wildlife of interest and what information we want to collect in order to decide exactly what it is we want to count or how to count them. So that brings me to my next point, which are the general guidelines to field methods in wildlife studies. So when we think about these general field methods, we want to meet and address 4 basic components. One is, what is our question of interest? We need to know what it is that we're asking in the first place so that we can understand what type of estimate that it is we're trying to quantify. Are we looking at a population or are we looking at a habitat? What is it that we're asking? So then the next general guideline is to collect data that will answer your question. And you're probably thinking, well, duh. But it also comes down to the fact that you don't have to collect everything. You want to try and target the data that is important for that question that you are answering. So for instance, if you're going through and trying to do a bird study where you want to understand what habitats birds prefer in a particular area, you could certainly go through and measure things like air temperature. You could look at weather. You could look at what types of vegetation exist where. You could look at what other animals are in the area. Ultimately, in order to make a decision on which data is relevant and which is not, you have to refer back to your initial question. Next, or point number 3 for our general guidelines, is that you need to decide on the desired sample size and acceptable error before you start taking these measurements. And the reason that is it will allow you to make more robust conclusions at the end of your study if you know how many data points you need in the first place. If you go out to explore a particular site and are only able to collect a small number, then you may not be able to make the conclusions you were hoping to or answer the question you were hoping to at the end. So knowing what it is you need before setting out to do it is a really important piece to this. And that also brings us to our last point, which is never to confuse statistical significance from biological significance, meaning garbage in is garbage out. And this is a really important point when it comes to what we're dealing with in days like today where we have big data. Oftentimes, we will get a significant result at the end of a statistical study simply because we have so much data that we're working with. However, that does not mean that there's a biological relevancy to that statistical significance. So when you're handling the statistics associated with a data set, you need to be very intentional about how you're approaching it. You need to know that the statistics that you're running or the questions you're asking of the data can be answered and answered in a way that make sense. So that brings us to what it is when we want to actually set forth and study a population. How do we go about collecting data in order to understand how many individuals are in a population of interest? And there are 3 ways to do this. There's a census, there are estimates, and there are indices. So we'll start with a census. A census is a complete count of a population. So as you might imagine in practice, this is very difficult and only occurs when we have endangered species or a tremendous amount of resources and effort. So endangered species because there are so few of them oftentimes, or a tremendous amount of effort because if you think about wildlife, they move, they hide, they are in a variety of areas that might be inaccessible for someone to observe. However, you can get counts on a limited number, especially if they have a pattern in behavior that allows them to be counted in their entirety. So for instance, if you have some island species where they are relegated to one island and are not able to move off of that island, or if they have a behavioral pattern that brings them to one location time after time, so that you know all of the individuals in a population will be passing through one point at one time in their lifespan. So because that is not an easy way to going about estimating a population or counting an entire population, we oftentimes rely on population estimates. So that's a more common way of approaching this. We end up setting a time and or spatial boundary, and we do our best to get a count of a subset of the population within those parameters. And then we have ways of using those estimates in order to understand the larger population. So for instance, we could use a mark recapture study like you've talked about, or like we've talked about in previous aquatics lectures. We could subset a population or subsample a population in a number of different ways, assuming that it is some sort of standardized level of effort or standardized time period or both. So as you might imagine, because there are a variety of ways we can handle this, there are also a variety of ways our estimates can be impacted. So we have to understand that it is just an estimate and that there is no way for us to actually capture every single individual, which means that's the biggest difference between an estimate and a census. We know there's going to be error in an estimate. We are accepting of that, and we likely know where that error is going to come into play so that we can make better judgments on, or better, I have a better idea on what the overall population number is likely going to be. Now, working from that estimation, if we do this in a number of years in a row, let's say, some regulated time period, if we do this multiple times, we start to develop something called an index. An index is a long-running estimation of a population. And so the National Breeding Bird Survey Program is a prime example of the power of indices and the important data that they can provide in terms of both monitoring and conserving wildlife. So in this case, the Breeding Bird Survey has been conducted annually since 1966, and it's driven by citizen science, so like people like you guys, collecting data roadside along about 25-mile routes, and those routes are all throughout the country and into Canada. So this volunteer movement has been exceptionally powerful at documenting bird population changes. And so let's talk about that a little bit more. These data have been invaluable in terms of documenting the distribution of species like that scarlet tanager that you see on your screen right now. And with all this data, we can examine trends over time. So for example, the northern bobwhite we see in this cute little picture here. He looks a little bit like a pheasant. Well, the northern bobwhite we've been monitoring since that 1966, and we're starting to see this big swath of red. And that's not good. That means that we are seeing declines in the entire population, pretty much the entire population. And this loss in population really signifies for us that there needs to be management attention driven towards this particular group, or this particular species, I should say, not group. And so it allows us to quantify how these populations have changed over time because of those repeated measures, because of that index that we have of this species and of others very similar. Using this data, then we can evaluate a model to understand what drivers are causing those changes. And when it comes to estimating populations, this is where we really need to see repeated samples. When we understand how a population is shifting over time, it can give us more of a sense of what it is we need to be doing in order to protect that species from further decline. And in this case, a northern bobwhite unfortunately gives us a good example of that. When we're thinking about estimating a population via one estimate or building an index, we know there's going to be some amount of error. And it's really important that we can quantify and evaluate this error. So if I were to talk about error, I would break it down into three particular groupings. As a review, we can think about accuracy. I'm sure you have all learned about that in a basic science or basic statistics class. There's also precision and bias. So accuracy, as I said, is a little bit of a review. It's how close an estimate is to a true value. So you can see here we have an estimate that falls right inside of that green true value. So we know that this is a highly accurate measurement. Whereas if we look just a little bit outside that true value, we also have an estimate that's a little less accurate. And if we move all the way out here, this little guy has no idea what's going on and he's incredibly inaccurate. But we can also talk about something called precision. We do really find confusion exists between these two pieces of terminology, so bear with me. Precision is when we're dealing with how repeatable an estimate is. We don't want to think about how correct it is, we want to think about how repeatable it is. So as you see in this picture, all of the estimates are clustered together on nearly the same number. So this is a very precise estimate, but it's not exceptionally accurate because it's not within that true value. We can also see an example that is not precise. You see these estimates are all over the board when it comes to repeatability. So we're not landing on the same value each time, which means that these are imprecise. Now, let's talk about the idea of bias. Bias is kind of how precision and accuracy relate together. And our goal is to avoid bias, which is a systematic error, or basically an error that has a pattern to it. So if you look at these panels, 2 of these are precise and 1 is not. However, that precision is clearly a pattern that is nowhere close to the real number. So if you look at these 2 clumped areas, we see that they are away from the true value. And they are away from the true value in a pattern. They're all landing on something in the same general location that is not the accurate or true value. Whereas if we are in the first panel, we see that this estimation is kind of all around that true value, meaning there is clearly something that we're missing, but that error isn't systematic. It's not a pattern that's making us arrive at the wrong conclusion over and over again. So let's just say, for an example, to give you guys an idea, let's say we're trying to count the number of deer in an area. So we lay out corn in an area to attract them, and we count any deer that takes the bait. But it just so happens that we picked a site that is next to a loud highway. In our study, we are only counting those that are brave enough to come to the area to get the corn and have a preference for corn in their diet. So are we totally missing those deer that are too skittish to come through the loud noise? Or maybe they aren't attracted to corn? And how do we account for these individuals? We're clearly biasing ourselves towards those that are bold enough to come into the area. And in my opinion, this table sums things up pretty nicely. As you can see, bias is in the top 2 panels. So we're seeing here is this true value that we want to reach. And here we're seeing one that is precise. They're all nicely clumped together, but they're nowhere near the true value. And this is imprecise, where panel 2 is imprecise, where we're seeing we aren't arriving on the same data point approximately time after time, but it's still in a general pattern away from the true value. So there's something that's making us go towards that area in a systematic or a pattern-like way. That drift is bias. Where if you look at the bottom panel in point 3, that's kind of where we really want to be. It's highly accurate and it's highly precise, meaning we can repeat the experiment and get the same number time and time again, and it happens to be the true value. And then in panel 4, we see an example that's not necessarily as accurate. We don't see the bias we did in the top panels because our estimates are kind of all around that true value. So with that, I'll leave you with the idea that balance is being able to repeatedly measure individuals and avoid that bias component, and that's really key when it comes to wildlife population methods and monitoring.